Hi everyone. I hope you are doing well in your ongoing preparation for the AP exam. I did want to review a couple other things that had been on your list of wishes for me to look over with you. And so we're looking at mitosis and meiosis. And these are two different things. They were brought up by students. So mitosis is, of course, part of cell cycle. Meiosis is more along the lines of reproduction, genetics. But because they are similar and yet different, I wanted to talk about them side by side so we can make sure to clear up any misconceptions that might exist. Okay, so let's just kind of go over that. We'll start with mitosis, and then we'll talk about meiosis and how it differs. So in Biology 1 in Texas, we have to know the stages of mitosis. So, and in addition to the cell cycle in general. So you have to know, prophase and metaphase and anaphase and telophase and what's going on in each individual stage, etc. In AP, you don't have to know the names of the stages, but you need to know in general what's going on, and like the purpose and main ideas, big concepts, processes, okay? So when we're thinking about mitosis, we're looking at a parent cell that's making two identical daughter cells. So you're going to have 2N, meaning you got one set of chromosomes from one parent and one set of chromosomes from the other parent, making you have two sets of chromosomes. So it's 2N. It uses the little n just like you use in math for some random number, right? So in our case, this is what the number means. And then the concept of being 2N, of having two sets of similar chromosomes, is called diploid. So remember, the di is for two, and then ploid is referring to like chromosomes, okay, or sets of chromosomes, really. This process makes somatic or body cells. So this is what's being used in your body to make all of the cells except egg or sperm. Okay, it's ongoing. It happens from the moment of conception, so fertilization starts the process getting ready for mitosis, and it starts from that first division until your death. Your body will be undergoing mitosis through that whole time. It is important to remember that a cell is only going to undergo the S phase once in cell cycle. So the S phase, of course, is where the DNA is replicated. So when we're looking at this picture, and it might be a little bit tough at that distance, let me see if I can make it a little bit closer. You can see, there we go. You can see the X's in the parent cell, and then the individual pieces in the daughter cell. So of course, reminders that is because the DNA has replicated. We still consider this one because this information and this information are identical to each other. It's like having a spare copy of something. It doesn't give you any benefit other than having a spare copy. It's not providing you more information. And so that would be something to be kind of aware of, okay? The only thing that gets separated, and the separation happens at anaphase, okay? So just metaphase in the middle, and then Anna is pulling away. It's like, go away, Anna, from our Frozen movies, right? So we pull that away. What's being pulled away in mitosis are the sister chromatids. So these are identical to each other, and then they get pulled away to make two identical daughter cells. If you have a malfunction in a mitotic division, it can lead to a damaged cell. That does not necessarily mean that the whole organism dies, but the cell may die, or maybe not. It could be functioning. It could be functioning, but just not at ideal circumstances. Or this could be the type of division that leads to a cancerous tumor. So that would be something to consider. So we're looking at errors in the cell cycle, Frequently, those are going to lead to things like cancer cells, but there are other possibilities as well. Okay? On the other hand, we're looking at meiosis. In meiosis, as we can see by this picture, we start with one parent cell that goes through one division to make two, and a second division ending with 4 daughter cells. And again, let me bring it a little bit closer so you can see the pictures just a little bit better. Okay, so you can see that you've got 4 copied chromosomes in the parent cell. In the first division, you get 2 cells, and each of them only have 2 kinds of chromosomes. And in the last one, you end up with 1 copy of each of those kinds of chromosomes. Okay, so that's like generally what we're considering. So you're starting as 2N, or diploid, but because you are dividing, you're not going to end up with the same number of chromosomes, you're going to 1N, which is called haploid. So hap, like half, thing to remember. This is the process by which gametes are made. In many organisms, we call these egg and sperm, and so organisms are typically genetically predisposed to make egg or sperm. In some organisms, like plants, they might be designed to make both. So in a flower, you may have both male and female parts. Just as in mitosis, meiosis only undergoes S phase once. So you start with the original 46 chromosomes in humans, and that doubles, but not really, right? It just makes a copy of that. So this is still considered 2N. So that would be your S phase. Okay? And when they separate, which happens twice, right? The first separation, we're separating homologous chromosomes. So if we were talking about, chromosome, let's just say chromosome 1, right? I got 1 chromosome 1 from my mother. I got 1 chromosome 1 from my father. Now, they won't have exactly identical information on it because my parents are not from the same, they don't have the same genetic background. So they're from different cultures. They have a variety of different genetic markers. So they're not going to be identical, but the genes will be the same gene. They'll just have different alleles, okay? So what that means is in the first mitotic division, that original cell, which is like a proto-oocyte in the case of females, so my proto-oocytes, which I was born with, inside the ovary, preparing for ovulation, would take that information and they would separate those 2 homologous chromosomes. So my mom's chromosomes, 1 would go in one cell, and then my dad's chromosome would go in another. Okay. In the second division, the sister chromatids divide. So remember that while these are done, they are connected. And so you have the copied mom chromosome over here, and it's going to divide in the second division. And then the copied dad chromosome over here, and it's going to divide in the second division. So some things that make this a little bit different and incredibly significant when you're thinking about evolution and genetics and all of that and how it ties together for helping an organism survive. We have this amazing thing called crossing over, which is fantastic. When it happens right, it is so instrumental in increasing genetic diversity. So basically what that means is during prophase 1 of meiosis, during the very first part of meiosis 1, the first stage, so this one right here, when the homologous chromosomes, so mom chromosome, dad chromosome, are kind of hanging out in the same area, getting ready to divide, if they overlap at all, they kind of cross over, we call that a chiasmata in that area, they're crossing over, they can swap genetic information. So then when the sister chromatids, so these are my mom's sister chromatids over here, Maybe this chunk is the chunk that crossed over from my dad's. So this part of the chromosome is my mother's, but this part is my dad's. And then this one is all mom. Dad over here, this one is all dad. This is the part which overlap with my mom. So this is dad part, mom part. And so they can divide. This can happen at multiple places in multiple chromosomes throughout the parent cell. And so we're going to see a lot of variance. So here's where you could get a little bit more of one parent than another in the egg or the sperm cell, okay? Or things like combinations that aren't, are a little bit different. So you got your grandmother's eyes, but your grandfather's chin. Something like that, okay? And that is definitely going to increase genetic diversity. So if we're talking about all of the egg or spermatogenesis, all of the sperm made at one time, if we were looking at an organism of a parent that was having twins or multiples, right, that are not identical, so they're fraternal twins, that would mean that in, you know, a division of those sperm, for example, you could get one sperm cell from a division that is mostly the baby daddy, right? So the man's sperm, that man, could be more of his mom's side and less of his dad's. And then another sperm cell, same division, can be more like his dad, but have a little bit of his mom too. And so your offspring, even though they came from the same man, could look very different. And that's what's going to increase that diversity. So siblings don't always have to look alike. And then when we're thinking about what could go wrong, which of course is going to be a very negative concept, when we're talking about meiosis, we're talking about what's going wrong in the production of gametes. So what happens if there's an error in making an egg or an error in making a sperm cell. So when we're thinking about this, this is going to lead to some sort of mutation in the offspring. That could lead to a variety of things. It could lead to the sperm not being able to function. So the sperm doesn't have a little flagella that can swim right. So that's going to decrease the likelihood of fertilization. It could also be something where the resulting offspring, due to this malformation or this malfunction in meiosis, is going to have an extra chromosome or a missing chromosome, non-disjunction, right? And that could be something like Down syndrome, or it could be like Creutzfeldt, or it could be like Edwards syndrome. And anything with a syndrome after it is where most of the people with that condition have similar traits. And that can be anywhere from upsetting to catastrophic when you're thinking about the viability of the offspring, the functionality of that offspring. These, when the malformation, there's the malfunction during meiosis is significant enough, it can also lead to what's called spontaneous abortion, which is miscarriage. And as you know, I've been through several of these. And they can be very traumatic for the parents, but they also can happen before somebody even knows that they're pregnant. So depending on what's wrong, if it's catastrophic enough, the fertilized egg may never actually embed in the uterus, or it does, but it can't reproduce beyond a certain point and will be lost. So it could be like a delayed period, or even a period that's on time. It's just such a short window, and it's not going to make it, which is a very depressing concept, and I'm sorry to leave you there. But when we're talking about mitosis and meiosis, it is important to think about like why it's done, where it's done, and what could happen if it goes wrong. Okay? So again, AP doesn't need you to know like in anaphase 2, no. But if it says 2, you need to know it's the second division, right? Not the first, but the second. And we think about what that's going to do. All right. If you have questions, and you might have tons, and that is fine, shoot me a text or an email. I'm happy to answer any questions you have. Best of luck, I know you're going to do amazing. I'm so proud of the effort that y'all have put in so far this year. Okay, I will talk with you soon. Mwah. Bye, y'all.